Once again, good afternoon, everyone. Thank you so much for joining. We're really happy to have you. I'm Abhishek from Proteus, and I'm really happy to have this opportunity to host the first talk of our 5-day symposium, Rhetor 2.0. And we really hope that you all have a thoroughly enjoyable time and tune in for all the talks and really have a great time. And our first speaker is the illustrious Dr. C.N.Ramchand. He holds a Ph.D. in biochemistry from MS University Baroda, where he also obtained his MPhil and served as an assistant professor there as well. He also has a master's degree in chemistry from the University of Kerala. He was a 3-time winner of the Young Scientist Award from Charing Cross and Westminster School, UK, for his work in the role of membrane pathology in schizophrenia and the development of EPA as a therapy for schizophrenia as well. He is currently the CEO and president of Saksin Lifesciences, as well as the co-founder and CSO of MagGenome Technologies. So today, we'll have Dr. Ramchand speak about the development of anti-VEGF antibodies against diseases like age-related macular degeneration, as well as diabetic retinopathy, and also kind of elaborate on the general process of drug discovery as well. So for the duration of the talk, I kindly request all of you to switch off your cameras and mute your microphone. We'll have a session to ask questions and interact with the speaker at the end. And I'd also like to mention that you can use the hand raise option and chat box to ask questions as well. So in a more general note, we are extremely grateful for the continued guidance and support of the School of Biology, IISER TBM, and also our faculty advisor, Dr. Sadananda Singh. And it is their collective support and guidance which has made this symposium possible. I'd also like to acknowledge our sponsors, MyBiome Therapeutics and JKG Biosciences, and also thank them for their support. So now we'll move on to what promises to be a really great session. And Dr. Ramchand, please.
Thank you. I'm just trying to get my screen here. Okay.
Yes, sir.
Can you see my screen?
Yes, sir.
Okay, it's just coming. Okay.
Yeah, we can see.
Thank you. Thank you for the kind introduction, and I'm extremely thankful to the team, IISER team, especially Mr. Siddharth Shivanand, who invited me for this program. I know I have about 45 minutes time to give this talk. Please remember that I'm just going to give a complete presentation briefly on how we have developed very first time a biologic molecule for a particular disease. So before that, I thought I will just give a brief about what exactly is drug discovery. Drug discovery, I normally give workshops. It's a 3-day program, but in 5 slides, I will have to just brief you. I will also give a brief about synthetic biology, which you all know, but that's what we have used developing our molecule. And this is the first time a molecule from India is developed, which has gone to what is called the Indian RCGM program, and we have reached the phase 1 in the US. And the molecule is now out licensed. So I'll briefly talk about that, how we have developed. So I will try my level best to as simple as possible. I know that you are students for bachelor's and master's. So and anytime you can ask some questions also. Now here you can see many companies which we have mentioned. All these are startup companies based in Chennai or Cochin. We have also a facility in Bangalore. You might have heard about MedGenome, which is one of our most successful company. The other one is MagGenome, which is myself is the founder and CEO of that company. It's a novel technology, magnetic nanoparticles or nanotechnology we have used for multiple applications. This is a very typical example of how university and industry can collaborate. The entire work has come from university, where I had several PhD students. But the most importantly, we have developed complete COVID kits like RNA extraction, RT-PCR, viral transfer medium, molecular transfer medium, and automation in this technology. And most of India, we are supplying this in the COVID time. So you can also see the other companies. CyGenome is based in Cochin, which is basically an incubation company, a sequencing company, next generation sequencing company. Saksin Lifesciences is the molecule what I am going to talk about, the development of an anti-VEGF molecule. Thiruvananthapuram Lifesciences is an ANDA company, abbreviated new drug application, or briefly you can say how to develop simple formulation for the US market. Thiruvananthapuram Biologics is another company which we are currently developing a molecule for diabetic retinopathy. We have multiple other companies are there. So today I am going to talk to you about drug discovery and synthetic biology, a brief about that, and development of anti-VEGF, vascular endothelial growth factor antibodies, FAB against eye diseases, mainly wet AMD, age-related macular dystrophy, and diabetic retinopathy. So I will briefly talk about the age-related macular dystrophy. Now, as I mentioned, the next few slides, I will briefly talk about the drug discovery. And basically, this can be divided into two. One is NCE, small molecules. The other one is biologics. Until now, most of the companies or drug discovery was focusing on small molecules, but it is shifting to biologics or, the protein molecules. Drug discovery is a very long process, probably about 12 to 17 years, and very expensive thing. That also I will talk to you briefly. In this slide, I just want to tell you what exactly is a drug, and what exactly that, how to explain a drug. So a pharmaceutical drug is a chemical substance used in the treatment. Either you have to treat a disease like metformin, cure. For example, you have to completely eliminate or and cure the disease like antibiotics, prevention, vaccines. You all know that the vaccines, especially how COVID vaccines are developed, or diagnosis. Many times, many of the molecules, drugs can be used for diagnostic purpose also. And the sources of drugs can be anything, especially animals, plants, inorganic materials. But right now, almost 80% to 90% of the molecules are synthetic in nature. That means the pharmacophore you will be developing based on the structures of the protein where it has to go and bind. And that is the most common method for developing the drugs thing. And there are various types of drugs are there. As per the US FDA, there are more than 400 classifications are there. Whether it is antipyretic, analgesic, anti-schizophrenic, antibiotics, like that, all different possible categories are there. But on the right-hand side, you can see, this is briefly the general classification of drugs. New chemical entity, otherwise a molecule which has got a patent can be called as a new chemical entity or as a new novel structure, which can be converted into as a drug molecule. Now, once that the patent period is over, that is called a generic molecule. For example, most of the molecules, what you are seeing in India, invariably can be generic molecule. That means any number of companies can be made. The difference in NCE is that can be only manufactured or licensed to another company or commercialized by a single company. The other one is biologics. Now, a lot of importance is coming to biologics. Like, for example, vaccines comes under biologics. Probiotics come under biologics. Or any kind of the molecule which is generally in protein in nature can be called biologics, which is otherwise recombinant DNA technology used products. Now, what is the complexity of drug discovery? So you need to understand it takes probably between 12 and 17 years. It takes about $2 billion. And there are about 35,000 human diseases are there. And I can say that 25,000 diseases we have no idea. On the other hand, few diseases we have absolutely good idea. For example, allergy, asthma, inflammation, antibiotics, or preventative disease like vaccine, there is a very good progress is there. On the other hand, many diseases, for example, CNS-based disease, schizophrenia, depression, manic depressions, Alzheimer's disease. Even now, we do not know even the mechanism. So this is the unfortunate situation. But we have very good idea about 100 to 200 good disease, but a reasonable idea, the mechanism of 10,000 disease, and quite a lot still we have to conquer, we have to understand. This is the basic situation. Now, how many basic drugs are there? There are only 2,000 drugs, 2,000 basic drugs, including what is called Me Too drugs. That means the structural analog of the existing drugs. Basically, 400, 450 drugs only is there. And as far as new chemical entities or small molecules are concerned, in the last 30, 40 years, probably few hundred drugs have come. And last year, only 7 or 8 novel molecules have come. The other thing is that, from NCE, there is a shift is taking place. The shift is from small molecule to biologics molecule. So this is basically the situation of drug discovery. The other thing is that you also need to understand how many pharmaceutical companies are there in India. India has got more than 25,000 pharmaceutical companies. But any of these companies have developed a novel drug? No. Hardly anybody, because it is very difficult to get a phase 1 approval in India. I assume you are able to hear, no?
Yes, sir. We can hear.
Okay. So there are about 26,000 pharmaceutical companies are there, but hardly any companies have developed a novel molecule, which is very difficult to get a phase 1 approval. I was the vice president and head of Sun Pharmaceuticals drug discovery program, and we developed few drugs, but then we had to go outside India for the phase 1 trial. This is unfortunately, even now, the situation is like this. Most of the companies are trying to get the approval from mainly from US or Mexico or Australia or Singapore. These type of countries, people go and get the approval. So this is briefly about what is the situation currently about the drug discovery program in India. Now, this single slide shows the complexity of drug discovery program. As I mentioned, it takes between 12 to 17 years to develop a drug. And normally, if you are talking about a small molecule or NC, new chemical entity, and if you are talking about absolutely novel molecule, invariably, and once you have identified the target is known. Roughly, this is the statistic says about 10,000 molecules has to be synthesized. When I was involved in developing certain NC, we had to synthesize about 5,000 to 6,000 pharmacophore to get into one single molecule. Invariably, because this has to go through a 5 to 7 year program before entering into the clinical evaluation. And the first requirement is a very valid biomedical hypothesis, which you all know. You should know the mechanism of the disease. What is the target molecule, which is invariably a functional protein, which can be an enzyme, receptor, ionic channel, or transport protein. And you need to know each and everything about that protein. Then only you will be able to develop the molecule. And it is very important that, the molecule needs to be targeted. If it is going and binding to an off target, that will be poisonous. The drug will not get approval. Selective to a specific condition, it has to be very potent. Earlier, 1 gram, 2 gram molecules also might be approved. Now you can see every drugs 5 milligram, 10 milligram, 20 milligram, that level. The molecule has to be physically stable. It has to be soluble. That means the molecule has to be reasonably water soluble, and so that then only you will get a better pharmacokinetics. Permeable, that means membrane permeability there. Metabolically stable, that means after reaching the bloodstream, it should not immediately convert it into its metabolite, otherwise it will be excreted immediately. It should not induce any particular protein or enzymes in the body. The binding of the drug to the receptor or the enzyme has to be reversible. The drug has to be durable. It has to be non-teratogenic, non-mutagenic. Finally, the molecule should be patentable. If the molecule can't be patented, it will not have any kind of value. Eventually, the molecule needs to be a manufacturable. The synthetic process has to be manufactured. Just understand. A single molecule, after 7 or 8 years, this has to go through all these processes, then only it will be successful. That's why it's extremely difficult process to develop a drug and reach the clinical practice, go through phase 1, phase 2, phase 3 trials, and finally a commercially viable molecule. These are the different drug discovery program, what is followed all over the world. And you can see from the beginning, it is comparatively simple. What is process development means? You have the structure, you need to synthesize that molecule, which is most of the companies are following, which is otherwise called API, active pharmaceutical ingredient is synthesized. Formulation development. Once you have the API, you have to formulate the drug, your tablets or the liquid or your injectable or your paste or emulsions. That's what is called the formulation development. Drug delivery system. You need to identify the way which you have to give as an oral molecule, as a liquid molecule, as an injectable, or a transdermal. So drug delivery system, you will be able to deliver through whichever is the route you want, but every time you need to get separate approvals. The other thing is that new drugs, how new drugs are how new molecules are developed. There are three different ways are there. I can't explain because of the time lag, but I will just briefly tell. Once your target is known and the pharmacophore is known, there is one way of synthesis. Many times the target will be known, you won't know the pharmacophore structure. So identify a novel pharmacophore. Unknown target, there are examples. The target is unknown, but the pharmacophore structure is known. You can analog the pharmacophore, still you will be able to do that. The other one is that biologics molecule. Then biologics process and biosimilars are made. Then biologics new use, metabolic pathway analysis. That means how a biologics molecule and novel applications can be come out. And the other one is that alternate use of existing drugs. So just briefly, I am telling about how various drug discovery program, which is followed by all pharmaceutical company. But the first few things are done in India, but no, I mean, hardly any company is involved in developing novel molecules or total new drug discovery program. And this slide gives about the drug discovery and development and the complexities. As I mentioned, there are two processes: discovery process and development process, and the commercialization process. And each part of this slide shows about the involvement of the scientists, what all things has to be done. Target discovery, that means how to identify the targets. Choose your functional proteins, DNA, RNA, any of these can be targets. Then lead identification and lead optimization. This is again comes under the discovery of the pharmacophore. Then comes that drug. Once you have identified the lead molecule, from lead molecule to drug molecule, which is called the drug development program, and which is divided into preclinical program, which normally takes about 4 to 5 years, which is followed by phase 1, phase 2, phase 3 trial, and finally registration and marketing. Now, simply you have to understand, it's an extremely complex process, takes 12 to 15 years, and approximately between 1.5 to about $2 to $2.5 billion is required for developing this molecule. Now, you all know about synthetic chemistry, medicinal chemistry, and process chemistry. That means a chemical structure can be synthesized. Now, using the same method, anything can be synthesized now. Now, what exactly is synthetic biology? Now, if you see about the cell, a cell contains outside cell membrane, which could be lipid in nature or glycoprotein. And inside, if you see, it contains DNA, RNA, protein, peptides, amino acids, and many carbohydrates, etc. So this is basically a structure of a cell, and the cell contains probably about few 100,000 different complex materials are there. Can we synthesize that? Yes, we can synthesize. If you see, you will be able to synthesize a protein, a peptide, a DNA can be synthesized using chemical processes. This is what we have done in our the synthetic biology process. What we have done is that synthesize the DNA. But then before synthesizing the DNA, we have used a technology called phage display technology, in which nucleotides are embedded in the phages or the virus, which we have used developing novel sequences for identifying better and better protein molecules. So just briefly understand, synthetic biology is just like medicinal chemistry or synthetic organic chemistry, where you will be able to synthesize whatever is the biological molecules, and eventually it is expected that a cell can be made, or the entire organism can be made by using synthetic technology. It can be using chemicals or other using enzymatic synthesis process. So this is very briefly, and there are multiple explanations are given for a synthetic biology process. But briefly, this is what, just like a chemical is prepared, the same way a biological molecule also can be synthesized, and how that can be used for various applications. So it's an emerging field of biology that aims at designing and building novel biological systems. The final goal is to be able to design biological system in the same way engineers design electronic and mechanical system. One interesting thing is that the synthetic biology came probably about 6 to 8 years back only, and back in 2016 about more than 500 startups started with synthetic biology process, and various applications are there. And left side you can see many companies are coming, is there, which are the synthetic biology company, and see the valuations of the company continuously going up. Currently it is expected about 40 billion is the valuation of synthetic biology company, and mostly these are located in the US. Now, what we are doing is that synthetic biology, you can say that, biology, engineering, and informatics combined together. That's the process. What we are doing is phage display technology. And using phage display, novel proteins can be made. And what we have developed is this human therapeutic, which is an antibody, a Fab molecule, fraction of antibody. That is what we have developed. So we have used totally synthetic biology process. Now, here onwards, I will briefly tell you how we have discovered and developed a superior anti-VEGF antibody for the treatment of wet AMD, or age-related macular dystrophy. It's a serious illness which is happening probably 40, 50 years plus, normally 60 plus years, losing the eyesight because of the degeneration of the macula or macular degeneration. Now, some of the facts about our company. It's a biologics company based in Tysell Park and established in 2014. And our key focus, development of antibody for treatment of wet AMD, age-related macular dystrophy. There are two different types are there. Dry AMD, which is the major type, majorly a large number of people are affected. The second is proliferative diabetic retinopathy and other diseases. Wet AMD, age-related macular dystrophy, that is a genetic abnormality, and proliferative diabetic retinopathy is because of the increased levels of glucose, or because of the diabetes, high levels of diabetes, and neovascularization takes place in various areas, especially in the retina. And our majorly we wanted is the development of either superior molecule or totally novel biologics. That is what our aim was, the company's aim was. So our main vision was provide affordable drugs for various diseases. So first started with macular degeneration, followed by diabetic retinopathy. What is the specialty of us? Saksin is having very strong expertise and experienced scientific team, but please remember this experience came through only training people. It was very difficult for us to get right people initially, because this type of work has very rarely happened in India. Especially we won't be able to hire anybody from other pharmaceutical companies. And we have all the equipments what is required for developing this. And our main thing was that developing novel IP. If novel IP or new IP is not there, the company will not be valued at all. And why? Why we have developed this molecule? Wet AMD has a global prevalence of 2%. It's a huge number. But 200 million people have got AMD. And is estimated to affect 196 million people by 2020, of which 10 million people were reside in India. Now, 196 is which is dry AMD or age-related macular dystrophy, which is not that serious, but about 15%, approximately about 30 million of this will be wet AMD. Now the problem is with the wet AMD, because if you get wet AMD or dry AMD, about 15% will be converted into wet AMD, which neovascularization takes place. The blood oozes out in the retina, and eventually you won't be able to see. Now, what are the major challenges with the existing products? Because the cost is very high, efficacy rate is not that great, because only 45% efficacious, and availability is, especially in India and developing countries, it is not there. So there are major challenges for that. Now, current anti-VEGF drugs are too expensive for cost-conscious market like India, UK, Canada. In fact, most of the countries, it's very, very expensive. When the first molecule, Lucentis, was introduced several years back, it was started with $20,000 per injection, and like that, 8 to 12 injections are required. So nobody can think about bringing such a molecule to India. But now it has come to about probably $500 to $1,000, which is still very, very expensive. So this was our first challenge, how to bring down the price, which we have done. Now we can even sell for less than $50 once we develop. That is what our calculation. And unless we bring the price, and if the drug is not better than the existing molecule, we won't be able to sell also. And we have used absolutely novel technology, fast display technology. This is not what the earlier technology, earlier Lucentis, which is also something similar to what we have developed by a very different method. The antigen, which is VEGF, was injected into mice or rat, and the antibody is taken, and which is further improved by using site-directed mutagenesis. And they took, instead of the entire antibody, they took the Fab molecule as part of the antibody, and that they have used. And I'll briefly tell about the wet AMD. Individuals above 60 are at risk. Global prevalence is about 200, now 200 to 20 million. And 10 to 15% is wet AMD, approximately 1/3 in India. So 10 million, which is 1 crore, almost about 1% of the Indian population, which is a very, very, very large. That means probably about 50 to 60 years, a very large number of people, maybe 5, 6, 7% people might be having this condition. It's a horrible condition because you have been seeing, but then suddenly you have started losing the vision. It is a terrible situation. Now, this is only one condition, that is wet AMD. But a similar way, diabetic retinopathy, it is much more. Those who have got diabetes, a large number of people after about 50s and 60s get diabetic retinopathy. Both these manifestations are something similar. Now, what exactly is the manifestation? Wet AMD is characterized by growth of new blood vessels in the macular region, loss of central vision. You can see this side, sorry. You can see this, I have given the eye structure. You can see macula, fovea is the central vision. And what is happening is that, after neovascularization, if there is, because of the genetic abnormality, vascular endothelial growth factor is more, what will happen is that, this produces lot of vasculature. That means blood vessel will be formed. After that, a large number of blood vessel comes, it breaks, the blood vessel breaks and the blood comes out. And if the blood comes out, initially the normal vision was like that, eventually you won't be able to see, or there will be spotted loss of visions will be there. VEGF is the key molecule that drives wet AMD. By removing VEGF, neoangiogenesis is blocked, leading to restoration of vision. So the idea is that such VEGF molecule, which is produced in high levels, has to be removed. How this can be removed? By using an antibody. So the antibody needs to be injected into the area, into the vitreous area where the retina is there, where the neovascularization takes place, and the VEGF is removed. This will not happen. So these are our company overview, our mission, our value, our key statistics, and IPR. So we have a complete protected IPR is there, intellectual property right. That's one of the most important requirements. So one of the other requirements is that you need to have a very dedicated team. We have about 30-35 staff for this particular company. These highly trained scientists from various areas. We have about 10 to 15 PhDs are there. So these are some of our advisors, and Dr. Sidhu is from University of Toronto. He is a synthetic biologist or a molecular biologist. The same is the case with Dr. Jason Moffat. He is also from there. Dr. Jayakrishna Ambati is one of the most famous ophthalmologists in the world. His brother, who got medical degree, MD from Harvard Medical School at the age of 15, you can just see. So Jayakrishna Ambati is one of our advisors, and Dr. Shekhar Sheshagiri, he was earlier with he was working in the US, and he is now one of our advisors. Now this is the structure of the anti-VEGF Fab antibody. You can see the right hand side. The right hand side you can see the full antibody, and this is the constant region. These are the variable region, and there is a heavy chain, there's a light chain, variable region, and the constant region. What we have done is that the approximate molecular weight is 148 kDa. On the other hand, here we have taken a part of the molecule which is about 48 kDa. This is what we have developed, and it is totally through the synthetic pathway. And in order to get a patent, because already something similar was existing, we need to have the CDR, a novel CDR, complementarity determining region, has to be developed. That is one of the most important things. Now, this is the process flow chart of the Fab 201 discovery to production.
Your video is not visible.
You can't see my video now?
No, sir, we can see you as well as your slides.
Yeah, because somebody came and told me you can't see. That's good. Now, this is the process flow chart, the Fab discovery and optimization. This is the very first discovery process. Again, a complex process. We have used fast display technology. Then the requirement is that, once you have the sequence, you have to clone it and transformation, and this is done in a bacterial system. Then upstream, that means fermentation process, lab scale, small scale fermenter, large scale fermenter. Then subsequent to that, you have to purify the protein. This is the intracellular protein, which is expressed over there. And bacteria produces probably 10 to 20,000 different proteins. It's very important to purify that particular protein into single entity. That's the most important thing, and multiple processes we have used. And other important thing is that after purification, it's not necessary the molecule will be stable in a condition, normal temperature. You have to formulate the molecule, formulate the protein. If it is not formulated, the protein will not be stable. So this is basically the discovery and the development step that we have followed. It probably takes about 1.5 to 2 years, and there is multiple analytical techniques has to be done. Probably about 20-25 different analytical techniques has to be developed. Now, this is the product development path. If you see discovery, Fab generation, Fab validation and characterization, Fab production, and in vitro and in vivo efficacy and pharmacology. Now, as I mentioned, there are absolutely about 20-25 different analytical procedures we have to follow, and also we will have to make sure the configuration and conformation of the protein. That means 3-dimensional conformation. That is called biochemical and biophysical characterization. And initial stage, we will have to do the proof of concept. unless the proof of concept is done. That means an animal model study we will have to develop. Unfortunately, this particular animal model, which was not available in India, we have to send it to the US. Then subsequent to this will go for scale up and regulatory approval. Now in India, it has to go through a multitude of regulatory processes there, getting the approval even to get into the animal experimentation. And you will go through a series of animal experimentation before going for phase 1. So before going for phase 1 or the first tolerance and safety evaluation in human being is done, the limited number of toxicity studies has to be done. Then comes the most important thing is that you need to produce the molecule in GMP manufacturing. The initial process is that material generation for clinical evaluation. You don't need to manufacture, you need to just synthesize or produce 1 gram of the product, formulated product, to do the initial 20 to 40 patients or human volunteers study. And that was one of the challenges we faced, because we went to Lonza in Singapore, we went to Switzerland, it was costing $10 to $12 million. We went to Syngene, which was costing $4 million. Finally, we have arrived at a collaboration with another GMP company. We could do it much cheaper. Now in India, it is called, the filing process. After the RCGM approval, you have to go through the filing process, Form 44. Now that number, that details have changed. It is Form 4. There is at least 6 to 7 approval processes there in India, just getting into phase 1. But before getting into phase 1, other approvals are there. But if you are going to US, there is 2 important filing is there, 1571, 1572. This is what is required for the US FDA. I can tell you comparatively much simpler. An ethics committee approval and a US FDA approval. In India, it is much complex, and somebody who is involved in doing will understand that. Now, these are the detailed step of discovery phase: Fab generation by phage display technology, Fab validation. You have to validate the Fab molecule and characterize it, identification of unique Fab clones and Fab production, and in vitro and in vivo efficacy. Any development process, initial process, when you have multiple clones, identify the right clone and make sure that better affinity, better expression, and all quality what it is required is there. And finally, you go for a simple animal efficacy study to make sure the molecule is working. Then you can develop it for further. The second is the proof of concept. So cloning anti-VEGF Fab, upstream processing, downstream processing, bioanalytical and impurity analysis. You can see the right hand side, all the different tests or analysis we have done. So you need to analyze and find out the sequence of the Fab molecules, DNA sequence, and it has to be transformation has to be done into the bacterial system. Then cell line development. Then there is a master cell bank. Then working cell bank. Then media standardization for the fermentation process. Then the fermentation, the whole time and other things has to be standardized. Then followed by cell lysis, the purification process and impurity removal, assay development, quantification of assay, then affinity checkup. So like that, a series of process has to be done at the time of proof of concept. Then the development process, formulation development, stability studies, biological activity, in vivo efficacy studies in rabbits, development of molecule and characterization. Now here the important thing is the stability studies. Stability studies means after formulating, if you are keeping it at 4 degrees, the molecule has to be stable over a period of at least 1 year. If the activity goes down, you can't use it. So every process, whatever we are doing is very, very important. Then biochemical, biophysical characterization. Because even after we are going for manufacturing, after formulating, at any time, the biochemical and biophysical characterization has to be identical. It is very important. A very long process is there to do this one. For example, PI determination, circular dichroism, intact mass. So a series of processes we have to do, and because of that insufficient time, I can't tell more in details. Please do understand what exactly is to be done is that after cloning, after the expression, after the fermentation, after the purification, after the formulation, the molecule has to go through all the analytical procedure to make sure that consistency is followed, whichever the batch you are doing, and at a later period also, after several months or years, it has to be identical when we are initially started with. Otherwise, you are not going to get the approval. So this is the consistency data which we have done. So that is like 5 batches we have to make, and the consistency, every batch is identical to each other. Whether upstream process, downstream process, formulation, everything has to be there, and 5 identical batches we have to make, and then we have to submit to whichever is the regulatory agency, whether US FDA or in India DCGA. And in India, for going for the preclinical study, you need multiple regulatory approvals are there. The preclinical studies in GLP lab, then approval from IBST for the submission of preclinical data. This IBST means in-house biosafety committee. So we have everything, which every month we will be meeting, and this has to be reported to RCGM very regularly. So that particular one, finally, once we get the approval, there is C1, C2, C3, C4, C5, C6. This is the process which has to go through review committee on genetic manipulation, or to Delhi we have to submit. And finally, we will get the approval for doing various animal studies. So the animal studies are dose range finding study, ocular irritation study, single dose toxicity study, 28-day repeat dose toxicity, single dose toxicity study in rats, and 28-day repeated dose toxicity in rats and rabbits. So 2 different species studies we have to do. What I want to tell here is that before going, how we are going to do the study, in which GLP lab, we have to get the approval process. Then comes the GMP facility. This is the material generation for the clinical evaluation. Then this regulatory submissions, and finally the clinical trial. So you can see that Fab201 is an efficacious anti-VEGF Fab molecule, which is a humanized novel anti-VEGF Fab. Humanized means if you are after injecting into the human, it will not produce any antibody. Is the other problem. If your sequence is not the right one, not humanized, what will happen is that this will produce antibody, then your molecule will not be efficacious. So if you can see right hand side that, you know, this is a cell-based assay. We have seen that, you know, our molecule is the most efficacious molecule compared to the existing molecule. So the Eylea, Lucentis, Avastin. Avastin is a molecule which is an anti-VEGF molecule, but which is used for the cancer treatment. But it has a different application. Some of the countries, Avastin is being used for treating wet AMD also. Lucentis is the first molecule, and Eylea is another one. Eylea is not an antibody. It is a receptor molecule which is cloned. So you can see this is the vehicle when we injected in an animal model, and this is our molecule, and this is comparative Lucentis. You can see how efficacious, whether it is Lucentis or our molecule, and you can see this. This is a VEGF-induced vascular leakage model. So it is very important to develop a very good animal model and make sure that, you know, it is working well. This is not a regulatory requirement. It is a requirement for the company in order to understand the molecule is going to work. And we have found that our molecule is more stable in rabbit ocular fluid also. Now, another thing what is required is that, you know, especially for the patent purpose and regulatory purpose, and the molecule is novel, you know, that paratop and epitopes. So the paratop or various CDR region molecule will go and bind to the antibody, to the antigen. Every molecule, which particular amino acid is involved in binding, that also we have to prove that one. It's a very long, complex experiment, which I can't explain at this point. And these are some of the functional assays for preclinical material used in the tox studies, affinity testing, surface plasmon resonance. So just understand that, you know, the molecule has to go through various biochemical, biophysical, characterization, and everything we had to do, and we have to compare with the existing molecules also. The other one is purity of preclinical material by SDS-PAGE and Western blot, so which you know very well. And you can see that if you see this molecule, there is a single peak can be seen. So this is using size exclusion chromatography. Another important thing what we have in our molecule we have found is that the formulation is robust and unique. We have found that this molecule is even stable at 37 degrees, which others could not do. And how this is stable, we do not know. So this is our cGMP facility, how the materials are generated, and our the research team only was developing this one. So this is finally what we have developed, and this is the one which will be used for the phase 1 trial. Subsequently, this will go for other batches will be developed for phase 2, other batches will be phase 3. Phase 3 will be done in thousands of molecules, but here you have to understand is that our ability to do is only up to phase 1. After phase 1, the cost is extremely high, several million to $1 billion. So that's the time it is given to another company, and in our case, we have a co-developer is there to go to the subsequent steps. There's another important thing you have to understand. I can't explain at this point, but when you are developing a molecule, before reaching the phase 1 trial, you have to go through a very large number of regulatory process. This is time consuming, and the regulator makes sure that, you know, when you are first time injecting, nothing happens to the human. That's what they are interested. So, but let me tell you, more than discovering, more than developing, to get the approval for the regulator is the most difficult thing. Now, what is the something like important? What we have done is that, you know, we have a very rapid turnaround time. We are going to a very large company. The time what is required to develop is very high because of the complexity of the other process. And because of a small company, we could develop this molecule in a very cost-effective manner. Now, I mentioned about eye diseases like macular degeneration. The other one is the diabetic retinopathy or proliferative diabetic retinopathy. There are other diseases are there which is mainly involved by vascular endothelial growth factor, which is diabetic macular edema, then macular edema, then choroidal neovascularization. And please do understand that, you know, about the cancers, solid cancers, many times because of the increased levels of vascular endothelial growth factor, neovascularization takes place in cancer, and because of that the cancer can grow high. So one of the treatment for many of the cancers are using anti-VEGF so that the blood supply to the cancer can be stopped. Now we are also trying for combination with other molecules too. But finally you need to understand that the IPR, intellectual property, now when you are telling it is very simple, but then we have 189 novel claims. That means right from the cloning process, the sequence, the upstream, downstream analytical process, we will be having multiple claims. We are making sure that nobody could repeat what we have done, and every company does the same thing. So it's very, very important that intellectual property protecting, and that's only for 20 years. But let me also tell you, it's a very expensive process, starting with invention disclosure. Subsequent to that, you will file a provisional patent, then patent cooperation treaty. By that time, you will lose 3 years, then followed by execution in various countries of your interest. We have done in 3 regions, which is North America, for example, US, Canada, then Europe, Australia, Japan, Korea, India, China. These are our countries. About 10 regions we have covered, which will cover probably 80% of the possible commercial opportunity. So I hope you have understood, and with this limited time, now I hope that in 45 minutes I have completed. But let me tell you, it's a challenge to complete in 45 minutes, whatever we have done in the last 5 years. But now you can ask questions to me.
So thank you so much, Dr. Ramchand, for really information-filled talk. We actually learned a lot about the entire drug discovery process, in 45 minutes, like you said, which is also quite difficult. And so if anybody has questions, you are free now to unmute and ask or use the hand raise option and the chat box. Thanks. And we'll try to keep it brief as well. Thanks. So in that case, sir, I think I'll start with the question. Is that
sure.
okay?
Yeah. Can you see me now? I can't see anything here. Let me see.
Oh, we can see you, sir.
Okay. So I don't know. Somewhere I'm doing something wrong.
Okay.
Yeah. That's okay.
So what I wanted to ask is there any way that you can, is this molecule specific to VEGF in the eye? Have you developed something to make it specific, or are you planning to look at it as a cure for like cancer or some other disease that you said, or inhibiting VEGF in other parts of the body as well?
Yeah, when you are developing a molecule, it is only possible to develop for one condition. So in this case, that our focus is wet AMD. We can't even go for another cancers or anything. But after first approval, you will be able to do much easier. So this is highly specific right now. What we developed, especially the formulation also, is for the eye condition.
So my kind of follow-up would be, how then did you make it specific? If you're targeting VEGF protein, how do you make it specific for the eye as opposed to some other region?
Right. Yeah, your question is valid, but then we are not injecting into the intravenously or anything. It is intravitreal injection to the eye only. And once it goes there, it remains there. So it will not have effect on any other place. So it is highly localized activity. But if you are talking about the cancers that you can give IV injection, and yeah, I'm just trying to see. I can't see anybody. So maybe we can come here.
Sir, we have one question in the chat.
Yeah.
So Vishal is asking, could you please explain how one can humanize an antibody? And he wants to know if you modify some specific sequences of amino acids, or do you add some amino acids along with the antibody?
No. Okay. Normal process is that, you inject an antigen into an animal, you will get an antibody. But then antibody is that, the two regions are there, CDR, as well as the constant region. Like, that constant region belongs to the animal. But in this case, what we will do is that, in the fast display technology, we have used the human, the basic sequences for the introduction in the phages or the virus. Okay, now I can see you people. There was a okay. So that is what we have done. Have you understood that? Then, so that means what we have used is complete humanized backbone, humanized DNA sequence for the backbone. That means the Fab region what we have used, which contains 48 KDA, approximately about 56, 58 amino acids, which is about like 150 the nucleotides are can be non-human. The other one is human. So because of the constant region which we are injecting and into the system, so that will not give any kind of problem. I hope I understood, and it is not because I could have given with the proper picture and other things to explain to you. So what we are I'm trying to do is that the basic the backbone of the Fab molecule what we have used in the fast display technology is the human DNA sequence, but what we are differentiating is the CDR region. So it is probably 98% highly humanized, but still some variation is there. Because of this, even after injecting this, antibodies can produce, and which is produced in our case also is produced, but certain level of antibody productions are allowed so that it will not completely neutralize the molecule.
Sir, another question I have is, how do you actually generate this peptide library for screening by PAGE display? Do you start with some known candidates, or do you generate a random peptide library and use that for your screening?
Exactly. Very random peptide library is there. So that is what we have used, and which is probably something like 10 raised to 12 random library, and which will be having an antigen, which is our the VEGF molecule, and the binding of the virus to that is used, and the virus is concentrated. Then like that multiple cycles are done. I should have shown the one of the slides how this PAGE display is done, so it would have been easy. Unfortunately, the time is limited. Because of that, I could not tell many of the things. I would be very happy at a later period if you want come, and each and everything can be explained. Your questions are very focused and specific one, but which I have not shown to you over here. I hope you people must have studied about fast display technology. The viral code protein and the DNA or the RNA in question will be embedded within the virus, and which will produce the code protein, which will be a novel one.
Okay, do we have other questions? Okay, in that case, Dr. Ramchand, thank you so much for your really informative talk. I think I can speak for everybody here in saying we really learned a lot. And many times doing research, we also don't think about, at least as students, we don't think about how tough it is to actually bring a drug to the market. And we wonder why there are not drugs for everything in the market. So I think that makes a bit more sense now. And once again, I'd like to thank the School of Biology and our sponsors, MyBiome Therapeutics and JKG Biosciences, for their continued support. Also, please tune in for the rest of the Rhetor talks. The next one is at 6:30 by Ms. Chinmaya Ganekar, and that's sure to be an exciting talk as well. So thanks everyone for attending. Thank you, sir.
Thank you very much. Thank you.